Everyone, this is Jenna. Welcome to week 6 of our unending spring break for 2020. I hope you all continue to do well and that you are staying safe, staying home, staying healthy, and keeping those around you healthy. So this week's little bit of a lesson is actually on SARS-CoV-2, sort of this specific virus that we're dealing with right now. What we know, what the urban legends are, the misinformation that's going around, and we're going to attempt to clarify some of that for you. So let's talk about the basics of this virus. What is SARS-CoV-2? SARS-CoV-2 is actually the name of the specific virus that causes COVID-19. It comes from the term severe acquired respiratory syndrome, coronavirus 2. The first SARS, severe acquired respiratory syndrome, which would have been coronavirus 1, was actually the first SARS outbreak that was observed in China in the early 2000s. And so this is similar in terms of symptoms and virology, and this is the second time or the second virus that this has been attributed to. COVID-19, on the other hand, is the name for the disease caused by SARS-CoV-2. It derives its name from coronavirus disease 2019. This is not the 19th coronavirus disease. It comes from the year that you can see here. COVID-19 and SARS-CoV-2 seem to get used interchangeably. Within the scientific community, you'll see a much more deliberate use of whether they are referring to the virus or the disease. In this lecture, I will try to stick with the virus name. However, you'll probably hear me refer to it as COVID-19 as well, and that's just the fluidity of our language. As you guys probably know by now, COVID-19 is really marked by a significant collapse of the respiratory system. Patients may start out with sort of a sore throat and a persistent dry cough, a fever, etc. But in a subset of patients, and in most cases, but not all, those patients are older, so over the age of 60, and may have underlying health concerns, or be younger, so younger than 60 with significant underlying health concerns, may progress to a form of full respiratory collapse, and that can lead to multi-organ failure. But the general current guidelines for symptoms include that persistent dry cough, a fever, and this is not just, a little fever. For some people, this is a fever that goes to 103, 104. Chest tightness and difficulty breathing. A subset of people report a loss of taste and smell. Body aches. Some people experience gastrointestinal distress, to include vomiting and diarrhea. In fact, the gentleman who was the very first identified case of COVID-19 in the United States up in Everett, Washington, did not present the way that we expect, and he instead presented with this GI distress and a fever. The fevers can lead to hallucinations. There is an overwhelming fatigue that has been reported, and a subset of people develop a severe headache. So where did this virus come from? Excuse me, I have allergies. So I've seen a bunch of urban legends and conspiracy theories circulating that the virus was manufactured in a lab in the United States or China or Russia, that it was developed as a bioweapon, and it was released in Wuhan or escaped the lab in Wuhan because Wuhan is a biotechnology hub in China. Here are the facts. We have sequenced this virus and we've analyzed it 6 ways from Sunday, and it is a normal, what we call zoonotic transmission, which means that this is a transmitted virus or infectious agent that moves from animals into humans. And this is something that actually occurs with fair frequency, and that it likely occurred from bats. The genetic sequence of this virus is highly similar to a set of coronaviruses that are found in bats, and it looks like that is where this may have derived from. In terms of tracing back to the initial cases, these people appear to have in common a wet market in Wuhan, China, where animals such as bats and pangolins and other things were actually kept living in cages to be sold as food. And it is likely that people were in close contact with these animals, and at some point the virus jumped from bats into humans. When a virus makes that kind of jump, we call it viral spillover. And this is a fairly common thing that happens in nature. So what do we know specifically about our SARS-CoV-2 virus that we're dealing with right now? For those of you who watched the lecture on viruses I posted last week, we know that this is an enveloped virus. That means that the capsid that protects the nucleic acids inside the virus actually comes from the cell membranes of the host cells. So this thing is a little bit like a Trojan horse. You have a virus protected by cell membrane that came from the host cell. Now, the virus does actually decorate the surface of that envelope with its own proteins, and you can see these proteins marked here. There's the spike glycoprotein. Remember, a glycoprotein is a protein that has sugar side units all over it, and those sugar side units are sometimes important to the immune system in terms of recognizing self versus not self. There's the E protein, and there's an M membrane protein M. And these are all little things that are put out on the surface of that envelope to sort of allow the virus to either evade detection or potentially to interact with the cells that it's targeting. We also know that this is a single-stranded positive strand RNA virus. Positive strand RNA viruses are particularly problematic because the RNA, once it is into the host cell, can immediately undergo translation to make more viral proteins. And so the rapidity with which it hijacks the host cell machinery is astonishing, and it also leaves us fewer avenues to interrupt that infection. This RNA, when it enters the cell, is actually relatively smart. It's small. It's only about 30,000 bases long, and it encodes a series of proteins, a small number of them, but those proteins are able to completely take over the cell. Okay, so how does it infect us? Well, we have heard through the news and everything that it is transmitted via small droplets of liquid, either from coughing, sneezing, or speaking. And those of you in class also know that right before we went on our extended hiatus, there had been data that I told you about showing that there is a significant oral fecal transmission because it is present in feces or poo. And that actually comes from the fact that this virus specifically infects cells that express something called the ACE2 receptor. ACE2 is angiotensin converting enzyme 2. You may have heard about that in anatomy and physiology if you've taken that class. But angiotensin converting enzyme 2, ACE2, has receptors waiting for it on specific cell types, most commonly cells in the respiratory system and in the GI system. The virus actually exploits those receptors. It binds to them, and when it binds to those receptors, it tricks the cell into taking up the virus. Basically, the cell thinks that angiotensin converting enzyme 2 has bound to the receptor and it needs to take it into the cell, but the virus has occupied the receptor and it's like a Trojan horse. It gets taken into the cell via endocytosis. It unsheathes the RNA and then almost immediately hijacks the cell's translational machinery to begin making viral proteins. Okay. I've heard a lot of people say, hey, I think I had this last fall. There is emerging data that is a little unclear to us right now. The genetic data and initial contact tracing actually suggests that the virus originated with 3 particular cases of this unusual pneumonia in Wuhan, China, in mid-November. That's the earliest date they can trace it to. And there are many coronaviruses. There are 7 that have been evaluated in humans, are known to infect humans, and some of these do produce respiratory symptoms. And so it is possible that SARS-CoV was circulating before that, but we don't actually at this point have data to confirm its presence in the human population before November 2019. Now, let me throw in a caveat to this statement. In the last 24 hours, there has been publication of information that there were initial deaths related to COVID-19 in California a couple of weeks before the very first acknowledged death here in Washington State, which was considered the first death in the United States due to COVID-19. That testing was done after a review of cases of unusual pneumonia-like diseases that killed people. If that is the case, and that would suggest quite significantly that coronavirus was actually circulating in the United States earlier than we expected, it is possible that, yes, this was here before we thought it arrived in January, and that there are a subset of cases that need to go back and be tested to see if perhaps that pneumonia was related to this. Okay, so what can you do? Well, you guys are doing a lot of what you're supposed to. 1, wash your hands. Why is this important? Well, you know, it's not just because it's clean and it's gross to like touch things with dirty hands and all that. And the fact that if you do have dirty hands, you can be leaving virus everywhere. But remember that enveloped viruses, such as SARS-CoV-2, actually tend to be more delicate. When you wash with soap and water, that soap or detergent actually disrupts that envelope. And remember, the envelope is made of lipid bilayer that comes from the plasma membrane of the actual host cells. Soap detergents tend to disrupt membranes. And so if you wash well with soap and water, that will actually disrupt the envelope, that will expose the capsid, and it is very fragile and will then be destroyed. So wash your hands with soap and water often. If you don't have soap, use hand sanitizer. A lot of you know I'm not a fan of hand sanitizer as a general habit. However, in this case, it is very, very effective. It does need to be 60 to 70% alcohol. If it is ethyl alcohol in the mix, it can be 60% or greater. If it is isopropyl alcohol, it needs to be 70% or greater. Now, why shouldn't you just use 100% alcohol, right? You can buy isopropyl, straight isopropyl alcohol at the drugstore. Why not use that? Interestingly, if we use a very, very high percentage of alcohol, that will actually do what's called fix the virus. It dehydrates it. And when it does that, it basically leaves it in a static state. It does not disrupt the envelope. It does not disrupt the capsid. And so if you fix that virus using a very high percentage of alcohol, you're leaving the possibility that the virus could be rehydrated later. Let's say somebody wipes off a surface or something and could become infectious again. So you're really looking for something between 60 and probably 75 to 80% alcohol, no higher. Don't touch your face, right? We talked about where these receptors are, and the easiest way for this to gain access to you is through your mouth, your nose, and your eyes, and especially in your mouth and nose, because they are part of your respiratory system. There are ACE receptors, ACE2 receptors close by, and that makes it easier for the virus to infect you. Practice social distancing. I know it's not fun not to like hang out with your friends and do other things, but staying at least 6 away from anyone who's sort of outside your immediate household makes sense. And in this case, you need to function as if you both are infected and not infected. You're trying to protect yourself from catching things from other people, and you're trying to prevent spreading the virus if you are an asymptomatic carrier. 6 feet away is the guidance, because that is generally when we look at the spew of small aerosol droplets or small droplets from people who are speaking, sneezing, or coughing. That is about the distance where they sort of will begin to fall. They go the farthest before they begin to actually fall to the ground. And so 6 feet away is a good guideline. And then please, new guidance suggests that you should wear a mask when you are out and cannot practice social distancing. Those cloth masks that we are all wearing right now, they will provide a very small amount of protection against inhaling the virus. If somebody near you has coughed or sneezed and not had a mask on, but they can significantly reduce the amount of virus that you spew out into the environment if you happen to be an asymptomatic carrier of SARS-CoV-2 or if you're actually ill. Okay, so that concludes this little information session. I'm going to post one later that's going to talk about how we study and detect SARS-CoV-2, a little bit about the serological assays that are coming online and what that means for all of us. As always, if you guys have questions, contact me through our Slack page or reach out by email, and I will talk to you guys soon.